Well, I think it really is a fantastic time in biomedical science. In the 30 plus years I've been doing this, I think the two most amazing methods, techniques that I've seen come about are the new way of sequencing, which is truly astounding. My very first paper sequenced 4 base pairs in a year and a half. Nowadays, in an afternoon in a machine in our lab, we'll sequence several billion base pairs. You just think about the order of magnitude there. And the other big, amazing advance is this new method of genome editing, we call it, to be able to go in and just change one little bit in the whole genome of any organism, a mouse, a human. And that's only about 3 or 4 years old and has absolutely exploded. Now, it's going to be a while, there are obstacles to overcome, but I think ultimately this is going to have a profound impact on the way we treat human diseases, particularly genetic diseases. And this technology has come about in the last 4, 5, 6 years. Before that, some of this wasn't even really imaginable. So I often tell my students, I'm a little jealous of them. This is a really exciting time to be starting out your career because there's so many amazing things we're going to do. And at first, it's going to teach us a tremendous amount, and eventually, I think it's going to impact the way we practice medicine. Again, this is an area where some of this same technology, particularly the sequencing technology and related methods, have revolutionized the way we do genetics. When I started out, we would look at one human variant a week in a few people. Now we can do 1 million at a time in an afternoon in 1,000 people. So it's totally changed the scale. And it's allowed us to do studies in tens of thousands, even hundreds of thousands of patients with the disease. And we're able to pick up little variations in our DNA that have an impact and allow us to begin to predict who's going to have this medical problem, who isn't. It's turned out to be more complicated than we thought. It's sort of good news, bad news. I mean, the good news is we're learning an amazing number of complex networks we never understood before. The bad news is it's pretty hard to dissect that down into information we can use today in the clinic to do this treatment or that treatment. I think ultimately this is will fairly soon, maybe 5 or 10 years, become a standard part of medical care. I suspect everyone will have their genome sequenced, and it'll be part of their medical record, and we'll use that to design all kinds of customized therapies and diagnostics, etc. But it's going to be a lot of hard work to get there. This is a big issue, I think, in medicine right now, particularly as we're getting more and more cost conscious and about the expense of medical care. And doctors in general, and I'm one of them, and I share that blame, we order too many tests. And I think we're just beginning to really think about this critically. When should we order a test? Just finding an abnormality, is that enough reason to order a test? And I don't think it is. I think we need to start thinking, is this test going to change what we do? One result will treat one way, the other result we treat the other way. That's a test that needs to be done. If we're going to do the exact same thing, regardless of the test result, maybe we should do it. And it turns out that a lot of the testing we do now for thrombosis risk in the clinic fits into that latter category, that it really doesn't change what we do. We sometimes create anxieties and concerns in patients that maybe we didn't need to, and we haven't always thought that through as thoroughly as we should. I think that's going to change, and this information will eventually probably truly guide the way we do treatment.